Hey, Science 10s. So this is biology, lesson number 4. So B4, transport across cell membranes. So this is a big one. You'll see that it's kind of the next 4 or 5 pages in your notes package. It's a long one. There's a lot going on within it. So I would say watch through this. And then just to get some of the visuals, you guys know again how I feel about Hank Green. Love it. This will be a good one for membranes and transport. This Amoeba Sisters, I think we watched one previously. Really good just for the visuals of what's actually happening. This one has a ton of views, so not as many, but I would say those two you probably want to watch. There's probably a Khan Academy one somewhere. This is part of the so passive and active transport. Good videos just for some of the visuals of this. It's a little hard to do in just a set of like a document. So I would have a watch through some of those videos. So just search membrane transport and kind of number 1, 3, and 5 that come up are good ones for that. So I would have a peek over those as well. So in your notes package, so transport across cell membranes, let's get going. I pressed record, right? Yes, I did. So full screen, there we go. So the cell can be described as a functioning open system that must acquire nutrients and excrete wastes to survive. So the word open system um, is something that exchanges both matter and energy with its environment. So it's pretty hard to have a closed system with anything. Um, but what it's saying is a cell is not an individual item that's totally self-sufficient. They need to communicate with others. They need to move things in and move things out. So it needs to be an open system with respect to both matter and energy. So you must learn about some of the major methods in which materials can be transported across cell membranes. So we've seen before in the last lesson, we talked about how something like sugar just can't find its way through the membrane. It's too big, so it needs to have another mechanism to get into the cell. So recall, cell membranes are semi-permeable, meaning that only certain things are allowed through the membrane. A membrane decides what to let into the cell based on particle size, as well as those proteins that are embedded within the membrane. So these proteins have a little bit of control over what's coming into or out of the membrane. So in order to study the cell's role in the cell membrane's role in transport, we need to consider the following definitions. So these are going to be important for our different types of transport. So something called Brownian motion. This is just the theory or the understanding that particles are in constant motion. So the particles within a solid are just kind of vibrating next to each other. They don't have the ability to move a lot. Particles within a liquid have a lot more space between them, so they're kind of swirling around and they interact with each other. And then particles within a gas have significantly more movement and they'll bounce off each other as they move around. But particles are in constant motion. Even within a solid, those particles are within motion, vibrating against each other. So selective transport. So only certain substances can cross a membrane based on its size, shape, and electric charge. So things that have highly positive or negative charges sometimes have a problem crossing the membrane as well. Concentration. So it's not just staring at something really hard. What concentration is talking about in terms of biology is, and in chemistry, I guess, describes the amount of solute in a solution. So what you could equate this to is somebody that likes tea a lot. Um, maybe a lot of people don't put any sugar in their tea. We'll do coffee, I guess. So somebody that likes coffee a lot might only put, half a teaspoon of sugar in their coffee. If they don't like it black, they put half a teaspoon in and they just have a little bit of sugar. Whereas somebody that hates the taste of coffee and is trying to mask it might get 4 tablespoons of sugar and just heap it in there. So the concentration of the one that has more sugar in it, so in this case, sugar would be the solute. Um, that's a higher concentration per the solution being the coffee afterwards. So that's what concentration is. And in Science 20, we do not a whole unit, but a whole portion in chemistry on calculating concentration as well. So a concentration gradient is actually the differences in concentration between two adjacent areas or two different solutions. So the difference in concentration between two adjacent, again, adjacent means next to each other, adjacent areas or two solutions. And so what it's saying is particles tend to move down a concentration gradient. So moving from an area of higher concentration to an area or a solution with a lower concentration. So that's saying that if I took those two coffee cups and was able to super carefully pour them in a cup that had a barrier in between it, like pour one into one half and one into the other half, and then remove the barrier, what would actually happen is those sugar particles from the higher concentration one would actually move, what we call move down the concentration. So the sugar particles would be moving across into the other one to try to balance out the concentration overall. So again, we'll move from an area of higher concentration to an area of lower concentration until it reaches something called equilibrium. So equilibrium will be related to equal concentrations. And so again, that would be like when there is an even distribution. Sorry, I have to write so big on this. Of molecules throughout a cell. Okay, so those are kind of some of those definitions that we need that will tie into all of our membrane transport. So there are two types of transport that can occur in a cell. So two main types, and then we'll kind of break them down a little bit further. So passive transport. So passive meaning it doesn't require a lot of energy. It doesn't require any energy. This is just kind of the natural flow. So passive transport, molecules move across the cell membrane and no energy input is required. So molecules move down the concentration gradient. So that's again going from high concentration to low concentration. Um, so usually I do just the most uncomfortable activity ever for this is usually what I do is I get everybody up And you know that little back corner of my classroom where I just pile all sorts of junk, and you guys don't even know all the junk that's back there. I usually make everybody go stand in that corner, and we get in really tight, and it's uncomfortable how close together we all are. And then I say, wait just a minute, guys. Like, I'm gonna go wait here. I'm just gonna go grab something off the printer. Give me 2 minutes. And so usually what I do is I go stand in the hall and giggle by myself because I'm a child. And by the time I come back in, unless you guys really love each other as much as you claim you do, what you've actually done is you've moved from that area of high concentration of students where you're all packed in too close together, and you've kind of started to flow back a little bit into the rest of the classroom space because you don't like that area of high concentration. You want to move to an area of low concentration while you're waiting for me. So usually you kind of flow away from that. The other thing that I do, again, because I'm mean and a child, is I go to the staff bathroom and I grab um, the whatever it's called, the room freshener spray. And then I stand up at the front of the classroom and I just spray like crazy. And it drives you guys insane because you guys are all sensitive to smells and you hate the stink spray. And so I spray, and at first I'm the only one that's dying and truly hating the smell of that. And then eventually the people that would be close to me, so thinking back to our class, whoever's sitting in kind of that first row next to my desk, you smell it next and it stinks and it's terrible. And then it kind of moves out from there. So it's going from an area of high concentration of the stink spray, stink covering spray, to an area of low concentration. So those people that sit farthest back are the last ones to really become affected by it. Um, so that's passive transport. So it's not really releasing energy, it's just flowing down a concentration gradient, going from an area of high concentration to an area of low concentration. So there are three types of passive transport. So stop thinking in the big picture now, start thinking in the cell. So the first one that we're going to talk about is called diffusion. Sorry, those are both F's if they kind of look like T's. So diffusion is just the straightforward movement of particles down a concentration gradient. So with my activity of you guys standing way too close together, you guys would kind of diffuse away from each other. So moving down a concentration gradient is again moving from an area of high concentration. I can't write it all there, of high concentration to an area of low concentration. And again, this movement is due to Brownian motion, so those particles vibrating against each other and eventually kind of bouncing off. So in cells, only fairly small molecules are able to diffuse. So things like water, oxygen, carbon dioxide. We'll talk a little bit about water in the next part, but the rate of diffusion is determined by cell size. So diffusion into the cell, or the rate of diffusion into the cell, is determined by the amount of surface area of the cell. And then the diffusion within the cell, so once things get into the cell, their ability to move around within the cell is actually based on the volume of the cell. So that's diffusion. Next one is osmosis. So osmosis is the diffusion of water molecules. It's specifically water, so water molecules. So from an area of high, I'm going to write high water concentration, so low solute, to an area of low water concentration or high solute concentration. So osmosis typically occurs through a selectively permeable membrane, which we've said the cell membrane actually is. So some important terms for osmosis, pretty specifically, but diffusion as well. So when we're comparing two solutions of unequal concentration, I guess this does actually have a little bit of equal concentration, but when we're comparing two solutions of unequal concentration, the solution with a higher concentration of solutes is said to be hypertonic. So for the T example, the higher concentration one would have more sugar. So solutes, again, is whatever you're dissolving in the liquid or the solution. So since it says have more solutes in them than cells do, so the result, as a result, water will leave the cell. So this is, we need to look at some of these diagrams to really make sense of it. So if we have a cell, and the concentration outside the cell for whatever we're talking about is hypertonic. Let's use lots of salt. The inside part, so this is our cell, we would call this hypotonic. I'll talk about that. Hypotonic. So less salt. So if we had, again, our selectively permeable membrane, and maybe whatever this salt is can't get through the membrane, but water can. So this selectively permeable membrane that we have, whatever it is now, won't let salt in and out, because we know if salt could go in and out, we know salt would want to flow down its concentration gradient. It would want to go from an area of lots of salt to an area of less salt. But if we can actually not transport salt across the membrane, so I'm just giving you an example here. We'll talk about more down here. So the membrane's saying no salt. It's selectively permeable against salt. But what can actually happen is this has lots of salt, not much water. This has lots of water, not much salt in terms of concentration. So what would actually happen is water would flow out. So it would flow from an area that has a high concentration of water to an area that has a lower concentration of water or a higher concentration of salt. So what would actually happen within our cell, if water is flowing out of it, it would actually shrivel. So that shriveling of the cell can cause plasmolysis. So can cause the cells to kind of break apart if it shrivels too much. So the solution with a lower solute concentration is said to be hypotonic. So I didn't talk about this, but hyper means more, hypo means less. And so if we had that same cell in reverse, and now it's like, I'll make it a different cell, lots of salt, not much salt. So if we had lots of salt here, not much salt here, again, if the membrane is selectively permeable against salt, what would actually happen is the water would flow into the cell because it's trying to balance out the concentration gradient. So water would flow into the cell to try to balance out the concentration gradient and make this quote unquote less salty by adding more water. So what it wants to do is reach equilibrium across that selectively permeable membrane. And so what actually ends up happening is if a whole bunch of water is going into that cell, well, that cell is going to start to expand. It's going to kind of bulge out, and depending on what's going on, if enough water goes into it, it can actually break the cell membrane apart. I like that it says explode. That seems more dramatic. So we're going to say that cells will just explode all over the place. And so that's the process called cytolysis, cytolysis. So lysis always means split. So the cytoplasm would essentially split open, break things. So solutions that have equal solute concentration are considered to be isotonic. So the word iso means same. So those have the same amount of solute as inside the cell, or across the membrane it has the same concentration. And so the net result would be water's moving in and out at the same rate. So predicting solute diffusion and osmosis. So each of the systems below has an impermeable membrane that can be removed. So we have a big tub and it's got something in between it that's separating this ended up off where it should be. This side should be distilled water. This side should be 5% salt concentration. So within that, it just has an overall percentage of 5% of salt. What's going on here? It says I'm getting a delivery right now from DHL and I don't think I've ordered anything. So this membrane is blocking in between there. So it says use arrows to show the direction of solute movement when the membrane is removed. So we pull this out and now we have distilled water and salt water here. So what would actually happen is, oh, I need colors. Whoops. I'm also going to take that out. So these little salt particles, what's actually going to happen once we remove this membrane, these salt particles are going to move over based on diffusion. And then our water molecules, distilled water, what actually would happen is water would want to flow this way in terms of osmosis because it has a high concentration of water, a lower concentration of water here because there's all that salt. So salt's going to be flowing that way, water's going to be flowing this way. So in this one, it has a membrane, and then what we see is we're going to remove that impermeable membrane. So we have 10% glucose on this side, and we have 5% glucose on this side. So glucose is going to flow this way because, again, higher concentration, lower concentration. So those glucose molecules are going to shift over to try to reach equilibrium. And while that's happening, oops, water is going to be flowing from the area of higher water concentration to the area of lower water concentration. Hopefully that makes sense what I'm doing. So I'm saying the red is the flow of glucose, the blue is the flow of water. I should have done blue over here too. And in this case, we have salt molecules, so they're the little circles. We have glucose molecules, they're the little triangles. They're both a 10% solution, so we remove this membrane or this barrier between them, and we'll see that our salt will flow this way. Our glucose will be flowing down its concentration gradient this way, and what would actually be happening in terms of water, in terms of osmosis, is it would be both directions. So water would be flowing in and out of both equally. So once that membrane is removed. So hopefully that makes sense. Again, those videos might be really quite helpful for some of the visuals of this. It's hard to see on like a little two-dimensional diagram. So on to, what is it? Oh, there we go. It says each of the systems below has a semi-permeable membrane that prevents the flow of starch but allows sugar and water to pass freely. So in this case, our membranes, we're not going to take them out, but it's kind of like the cell membrane in that it allows some things in and out, but not necessarily everything. So it's got a selectively permeable membrane. So it prevents the flow of starch. So starch can't get across, but allows sugar and water to pass freely. So in this case, again, sugar, we have our little triangles. So that membrane would allow sugar to move across. I guess I'll use blue for water. And water would move this way. So water would move from its area of higher concentration to lower concentration. Um, since this membrane for letter E here is impermeable to starch, these little starch molecules can't actually move that way. But what can happen is the water can move from an area of higher water concentration to an area of lower water concentration. So again, the movement of water is that osmosis. The movement of other particles down their concentration gradient, like sugar, that's diffusion. Last one is sugar and sugar. This one's a higher concentration though, 3% sugar versus 6% sugar. We know that sugar is going to flow down its concentration gradient. Oh, I was like, why do I have that written wrong? And then water would flow in this direction. So water would be flowing from an area of higher water concentration to an area of lower water concentration until again it reaches something called equilibrium. The next one, so we've gone through two of our passive transports. I told you this was going to be a long lesson. To our next passive transport called facilitated diffusion. So although small particles like water, oxygen, and carbon dioxide can diffuse easily through the cell membrane, other substances can't do this on their own. So particles in facilitated diffusion, particles are still moving down a concentration gradient. But instead of just moving across the membrane, it involves the use of membrane proteins, which are specific to dissolved molecules. And so it's believed that molecules bind to specific sites on the protein and then the protein changes shape, helping the molecule move through the membrane. So it's kind of like, oh, do I have these diagrams on here? Good. It has some examples of the type of protein that it would have. So this one, it's showing glucose. So there's a glucose transporter. Glucose has a specific shape of it. It's a ring shape. And so it would find its way into this glucose transporter. That transporter recognizes, hey, this is what I've been looking for all my life. It's glucose. And so the transport protein shifts to alternative conformation. So it locks in, this side closes, this side opens, and now glucose can be within the cell. So glucose is released inside, and then the protein says, bye buddy, I'm gonna look for more, and it switches back to that original conformation, looking for new glucose molecules. So it's just helping it through because that glucose can't fit through this membrane. It's too big. So it needs to find one of these transporter proteins to allow it to come in. Channel proteins are kind of like that. They're a little bit less specific. They just allow typically many different things through it. So particles like glucose and ions, so again, positively or negatively charged ions, are helped across the membranes by transport proteins. Again, these proteins are embedded throughout that bilayer membrane. So the two types are channel proteins and carrier proteins. So carrier proteins change shape, that's like this one here. for particles like glucose to be transported across. And channel proteins are more for ions, a little bit less specific. And it has a tunnel, like it's just like a little secret tunnel for ions to move. So ions have a tough time going across this membrane due to this hydrophobic region in the middle. Doesn't like that positive charge. So any ions that need to get into a cell typically move across channel proteins. Can I do this all as one lesson? I want so badly to pause it and get a glass of water, but I'm going to keep going. I got this. So passive transport, again, required no energy. And again, flowing down a concentration gradient from an area of high concentration to an area of low concentration. So now we're going to talk about active transport. So at times, transport by a carrier protein, I guess might be the more sensible way, protein carrier in the cell membrane requires energy because it's necessary to move particles. Oh, I am not going to have room. Against the concentration gradient. So this is when we're trying to move something from an area of low concentration to an area of high concentration. So the energy needed is produced in the mitochondria through the process of cellular respiration. So if you're taking Bio 20, you'll learn a lot about cellular respiration, which all of our cells are doing, big thing in the mitochondria, to produce that molecule called adenosine triphosphate, or what we usually shorten as ATP. Adenosine triphosphate. And so ATP is essentially the energy currency of the cell. It's what the cell spends for energy to move and grow and reproduce and produce proteins and all of those things. So it says, explain active transport using the diagrams below. So this is not what I wanted, sorry. So what it's saying is right now we have sodium here. Sodium, low concentration. And here it's sodium. But you see there's lots of them. Sodium, sodium, sodium. So that's high concentration. And what we actually want is we want more sodium to get into there. So we're moving sodium against its concentration gradient from an area of low concentration to an area of high concentration. And that actually requires energy. Ooh. To do, because it's not just naturally flowing. We're kind of putting something where it doesn't necessarily want to be. Is that all I have for active transport? Yes, so it just requires energy across that membrane to push something in that doesn't necessarily want to be there. So some materials are too large to move across the cell membrane, even if assisted by transport proteins. So some things we wouldn't be able to get a protein big enough to let particular things in. Otherwise, it would just be creating essentially a pore for anything to flow in. So what cells sometimes do, we need to use the process of endocytosis and exocytosis. Sorry, that's like almost impossible to read. ENDOCYTOSIS. Exocytosis. And so this ties into this part. So it says large molecules such as protein molecules and bacteria are molecules that are insoluble in water. Again, can't be dissolved in water. such as cholesterol, cross the membrane by using vesicles. So we hear a lot about cholesterol being bad for you. That's when it's in your bloodstream causing backup of your arteries by causing plaque and decreasing the diameter, making your heart work a little bit harder and a lot of pressure on your blood vessels. Cholesterol, though, is actually important within cells as well. You don't need to have tons and tons of it, but it's important within cells for cell-to-cell communication. So in endocytosis, so we have that up here. So endocytosis. So endo means enter. Exo means exit. Um, so in endocytosis, a cell takes in large molecules and particulate matter by forming a vesicle in the plasma membrane. So a small area of the plasma membrane or the cell membrane sinks inwards to form a pocket or sphere. The pocket deepens, it pinches, forming a vesicle, which transports its contents to other locations in the cell. So what this is saying is, here's my cell. It wants to get this little star inside, whatever the star is. And so it's not super easy because that star is so big. We don't want to just create a channel here to let the star in because then it's huge and anything can get into it. So what it does, this is going to be the worst series of drawings you've ever seen, is the star will come up beside the cell, and what the cell will actually do is kind of start to wrap its little happy cell body around the star. Until eventually it can kind of pinch off. So it will pinch off. So now the cell is surrounded by cell membrane here. The cell has closed itself off, and this goes to wherever it needs within the cell. So that's endocytosis. It's bringing something into it. So sorry, I'm just trying to see. So a vacuole may form from the vesicle, which functions to store the ingested material. So just a storage unit until the cell needs whatever it's looking for, it has stored within the cell. So there are three different types of endocytosis. So phagocytosis is endocytosis of large particles. So pinocytosis, this usually is cytosis, that S doesn't look like an S, of fluid particles. Or this last one, it's not, this is not written the right way. So receptor mediated endocytosis, what actually happens is receptors on the cell surface, these are essentially proteins from the membrane surface, detect specific compounds that they're looking for. So these proteins are essentially looking for particular things that they know the cell needs. It's looking around for them. These receptors identify that that's what it is, and they cause a change in the cell membrane that allows that particular thing to come in. Ouch. So there's a nice diagram here. Boop. So I think I just said, yeah, I just said fluid particles, also really small particles for pinocytosis. So these are the different types of endocytosis that can be taking place. So phagocytosis, so this is our plasma membrane or our cellular membrane. This, whatever this is, this solid particle that we want to get in, finds its way to the surface. That membrane pinches off around it, so it's going to pinch off around it, and now this solid particle is housed within the cell, and it's just kind of sitting there until the cell needs it. So whatever this is, they call it a food vacuole. This is essentially a cell kind of eating, for lack of a better word. Pinocytosis, so something really small finds its way over. So same idea, it's going to pinch off, form that vesicle. So the vacuole is something bigger. Vesicle is usually quite small. And receptor-mediated endocytosis, so these little receptors on the surface are looking for, I like that they use stars too, it's looking for whatever the star is, and then once those receptors have detected it, it changes the membrane, the surface of the membrane, and pinches that off, so a coated vesicle forms in that case. Oh, I don't know if you can see that. Let's see if I can make it before my computer dies. Sorry, it's not too far. Plugging in. Plugging in. Uh-oh, what am I hooked on? This is intriguing teaching you guys or learning you're getting right now. All right, back over here. Not sure if you saw that. It was threatening that my battery was going to die. Um, we don't need to talk about HIV stuff. You can read that, but that's just some of the interesting stuff between HIV since we talked about it a little bit earlier. So exocytosis is the reverse of endocytosis. So it's the cell trying to get rid of something, a waste product, or something that was produced within the cell that needs to be transported to other cells. So exocytosis is kind of the reverse process. So in this mechanism, the membrane of the vesicle or vacuole fuses with the cell membrane, kind of spilling the contents out of the cell. So just think about this happening in the reverse. This little vesicle finds its way out to the surface, they connect, and then it opens up and spits out whatever was inside that vesicle. So many secretory cells, so cells that secrete products. So if you think of something like a gland that's producing hormones, so that gland is producing hormones that need to go to another target gland or target tissue or out into the body. So these secretory cells use exocytosis to export their products. So so enzymes is one, so cells that are producing enzymes and cells that are producing hormones. Again, the cell is going to produce it. It's not really a waste product, but it's something produced within the cell, so the cell needs to get rid of it. Hormones are pretty big, so they can't get out through the cell membrane across that phospholipid bilayer. So there'll be a vesicle formed around them. They'll find their way to the surface and get kind of spit out at the side of the cell. So these processes of endo and exocytosis both require energy from ATP for the rearrangement of the cell membrane. So it requires energy to be moving the membrane like that. So it says fill in blanks on the diagram, so use the following terms. Cell membrane, solid vesicle that's for some reason missing the letter I, solute, phagocytosis, secreted solute, exocytosis, pinocytosis. You may need to use some terms more than once. I'm just going to pause this for a sec because it might be on your worksheet, so just let me check. Okay, I got my life together. So there's actually two worksheets that are going to be up. So there's one called Transport Across Membrane. So it looks like this guy. And what you're doing is just looking at this diagram. So A, B, C, and D. Tell me about what that type of transport is. And then, and not just the word, give me a little bit of a description of how that happens. And of course, diffusion and osmosis worksheets, the ones before. So you're going to do some matching terms for setups below, draw arrows indicating the molecules, and so read which is permeable versus impermeable. A couple different questions. Show me, draw the, for question 4 here, draw what's going to be happening in terms of movement, circling these ones or writing it out if you don't have them printed. So there's two worksheets for that. So again, diffusion and osmosis worksheet and transport across membranes. So I'll just zoom back to this and we can fill this out since it's not the same. This is again from that blue textbook, which we haven't even touched. We haven't even touched the red textbook, so that's something. So from the textbook, so again, this is that solid part that's trying to get in because this is an example of phagocytosis. And again, that's forming that vesicle or a food vacuole, you could call that. G here is the cell membrane. So F, this is for, sorry, for B, this is pinocytosis. And so this is like small molecules and fluids and stuff. So I would say this is probably solute, whatever's dissolved in this fluid, little tiny things. And then for C here, this is actually exocytosis because it's working in the opposite direction. This little arrow is supposed to give us that indication that it's going from inside the cell to outside the cell. And it's taking a vesicle and it's kind of splurting things out. So that's our secreted solute. Oh, sorry, all the terms are up here. So secreted solute. So again, exocytosis is exiting the cell or being released into the environment outside the cell. So it's heading that direction, whereas these two are bringing things into the cell. So that's that diagram. Holy moly, that was a long lesson. So those two worksheets, make sure you get those to me and we will chat at you guys soon.